Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PUC first and second year. If you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as video is getting uploaded. Okay guys, we shall start today's video. In our previous video, I have discussed about the structure of an ovum. So today we shall discuss about fertilization and implantation. This fertilization and implantation in your textbook, it is on page number 51. Okay, so we shall start with the fertilization and implantation that we shall see. Right. So first, fertilization. Fertilization is what? As you know, fertilization is nothing but fusion of male and female gamete. Okay, I shall show you here. This is a male gamete, and this one male gamete is nothing but a sperm, and a female gamete is nothing but ova. These are the two male and female gametes, right? Ova and a sperm. Now the fusion of these two, we call it as fertilization, right? The fusion of these two, which one? Male gamete and a female gamete, we call it as fertilization. Now in this fertilization, during this fertilization, what happens here is in human beings. The first step here it takes place is copulation. Okay, that is copulation. Copulation is also called as coitus. During copulation or coitus, what happens? During copulation or coitus, the male penis will release its sperms or semen in the female vagina. Okay, now this semen, what is semen? Semen which consists of seminal plasma and sperms, both together we call it a semen. Now this semen is released by what? It is released by penis. Penis will release semen where? In the vagina of the female. Okay, vagina of female. So when it is released in the vagina of the female, now this semen, it consists of sperms. Sperms also starts to move here. Now this process of release of sperms or semen in the vagina of the female is called as copulation. It is also called as insemination. It is also called as coitus. Okay, so different names have been given for this. If you go through the female reproductive system is something like this, right? With the fallopian tube, something like this. I am just drawing a rough diagram of that. Here we have shown vagina. As we have discussed the complete structure of female reproductive system, you can go through that diagram and you can find this one is vagina. Now in this vagina, what happens? In vagina, that penis will release that semen, right? That process we call it as insemination. So when semen is released in the vagina, sperms, they are undergoing capacitation. Okay, the second step is what? Capacitation. First one is copulation, second step is capacitation. In capacitation, as soon as sperms or semen have been released here, sperms will undergo certain changes in the genital tract of the female so that they will remain active and they will start to move. Okay, they will start to swim because sperm is having this kind of structure. Is it? It do have presence of tail. With the help of tail, with the whipping movement of the tail, it will start to move towards the uterus and then it reaches to the fallopian tube. Okay, we have two fallopian tubes. You can find one fallopian tube is here and another fallopian tube is here. The sperms can move to any of the fallopian tube. Okay, sperms will start to swim and reach the fallopian tube. In fallopian tube, we have shown here ampulla isthmic region. Right, ampulla isthmic region. Now what is this ampulla isthmic region? I will show you that one in your female reproductive system diagram, which is there in your textbook. It is here. Okay, so this was the vagina. In vagina, what happens? That penis will release the sperms and those sperms will undergo capacitation. They will get altered and they will remain active and they will start to swim through the uterus. This part we call it as uterus. Through uterus, it is reaching where? To the fallopian tube. In fallopian tube, you can find it is very clear. There is presence of ampulla and isthmus region. Now, ova will be released from where? Ova will be released from the ovary. It will enter into the fallopian tube and reach to the ampulla isthmic region. The region between the ampulla and the isthmus. So when it has been reached here, sperms also will move and they will reach to the ovary and they will get fertilized. This fertilization takes place only when both the ova and the sperm are released at the same time. If both are released at different timings, then fertilization chances are very less. So that's why every coitus does not lead to pregnancy. Okay, Fine. So this is that structure. You can refer this, and I have explained this structure in some other video. If you want, you can ping it to that videos. Fine. So we were here. This we are showing it as ample isthemic region, as we have shown. This is also ample isthemic region. Now ova will be present here. Okay, now ova is present here, sperm has been moved here. Now that ova I shall show it outside. Structure of ova already we have seen. If you can imagine, this is an ovum which is present in the fallopian tube in an ample isthmic region, and sperm is coming from the, that is the uterus. As it will come to the uterus, as it will come to this ova into the fallopian tube, it will start to reach here. Here we have shown the outermost layer, the corona radiata, right? The outermost layer of the ovum, we said it as corona radiata. This sperms will start to reach here, and sperm is having acrosome. Because of the activity of the enzymes present in the acrosome, those enzymes will dissolve this corona radiata cells, and even it will dissolve this zona pellucida layer, the thin transparent layer. That zona pellucida layer also will be dissolved. Now as soon as one sperm has been reached to this zona pellucida, to this ova, the zona pellucida will undergo certain changes so that no further sperms can get fused. Okay, here so many number of nearly 300 million sperms will be released during one insemination. We have said it. In that 300 million sperm starts to move towards the fallopian tube where ova is present. Now that ova, to near that ova, many number of sperms can reach. Now it is like a race. Now which sperm will reach first, that will get fertilized. Now we have shown this sperm has been reached first. So as soon as it has been reached to the zona pellucida layer, when zona pellucida has been dissolved, it will allow this sperm head. Sperm head, what does it consist of? It consists of acrosome, and even it consists of a nucleus. As soon as acrosome, it will be utilized for dissolving this corona radiata and a zona pellucida. Now what will be left? Only there will be presence of nucleus. Nucleus will be left inside. Now tail's function is also not there. Tail will be released out. What was the function of a tail? That it helps in the movement, right? When it has been reached to the ova, there is no use of now tail, so tail will be released out and acrosome will be utilized. Only nucleus will be left over. Now that nucleus will reach inside this ova. Okay, when it has been reached here, what happens? Zona pellucida will undergo certain changes. It will become thick so that it should not allow, okay, it won't allow further sperms to penetrate inside. Okay, only one sperm can enter inside, which leads for the fertilization. Because of this, we call it as monospermy. Monospermy means only one sperm is allowed to get fertilized. Okay, mono means one, spermy means sperma. Okay, only one sperm is reaching to the ova and getting fertilized. So after capacitation, there is the third step called as fertilization. In fertilization step itself, you can find there is a monospermy. Okay, now the other sperms could not penetrate because zona pellucida, it has got thickened and it has got certain changes which does not allow other sperms to enter inside. Now if I am showing, this is that ova, and that nucleus has been entered inside, sperm's nucleus, and this is the we can say the secondary oocyte. Now till now I was saying it as ova because to make you all understand clearly, but actually this will not be an ova. This will be an secondary oocyte. That secondary oocyte will undergo second meiotic division. When sperm will enter, then second oocyte will undergo second meiotic division to form what? One ovum, and another one will be second polar body. Okay, one ovum and second polar body will be formed here, which means this we have shown in your oogenesis. Okay, so here what happens? As soon as this sperm is penetrating inside, now that secondary oocyte will divide itself. It will complete its second meiotic division to form one second polar body and one ovum. Now that ovum's nucleus will start to fuse with this nucleus. Now this will be the nucleus of the ovum. Now both will start to fuse together. Okay, this fusion we call it as what? We call it as fertilization. Okay, this fusion is called as fertilization. The process of fusion of male and a female gamete, we call it as fertilization. Both the nucleus will fuse. As soon as both the nucleus has been fused, what happens? What it will result? It will result in the formation of zygote. Now zygote is haploid or diploid? Zygote is diploid. How it got diploid? because both the gametes were haploid. Each gamete was haploid. When both have been fused together, n plus n will get 2n. Hope you got. Now zygote has been formed. Now all these changes, where they are taking place? They are taking place here in the ampulla isthmic region. You can get, where does the fertilization takes place in female reproductive system? That is ampulla isthmic region. In an ampulla isthmic region, fertilization takes place. Now zygote is present here. Okay. Now this zygote will be moved from this region to the uterus. Okay. Now zygote, it has been formed. It is a diploid. It is a single cell. Because it is a single cell, that's why you will find that every organism has been formed from a single cell. Now the baby, the newborn, everything, it has been formed from a single cell. Now this is a single cell. Now this single cell, it has to reach to the uterus. As it starts to move towards the uterus from the ampulla isthmic region, it will start its division. Okay. Okay, I will talk about that. Before that, here, as soon as zygote has been formed, at this stage only you can determine the sex of the fetus. Okay, you can determine the sex of the fetus, which means at this stage, as soon as male and a female gamete will fuse, at that stage only we can say whether it is going to become a male or a female child. Okay. So how we are going to identify how it has been decided at this stage, that I shall discuss later. Anyhow, we can know what will be the sex of the fetus at this stage only. At this stage it will be decided whether it will be a male or a female child. Okay, as we were here, zygote, it has started to move in this direction. Now when it has started to move in this direction, that zygote, I am showing it here, this will be a zygote. Now the zygote will start its mitotic division, simple mitotic division. When it has started a simple mitotic division, then what happens? This single cell will become two cell. Okay, two cells have been formed. It is diploid, right? Now because it is undergoing mitosis, that's why both the cells will be diploid. Now each cell in an embryo is called as blastomere. Each cell. Now how many blastomeres are there in this stage? 2 blastomeres. Okay. When zygote has started its division, the number of cells which has been formed, each cell we call it as blastomere. Now 2 blastomeres are there because it is undergoing mitotic division, so both are having equal number of chromosomes. After that, again it will divide and forms 4 cells. Again it will divide and it forms 8 cells, and it continues. Okay, and it continues and it shows many number of cells. Now when it is having 8 to 16 blastomere stage, okay, when the embryo is of 8 to 16 blastomere, when this embryo is having 8 to 16 blastomeres, for that we call it as morula. Okay, this morula will be of mulberry shape. Okay, this one can be, you can get in an entrance exam. Okay, so morula will be of mulberry shape. What is morula? When a zygote, when an embryo is of 8 to 16 blastomere cells, then it is called as morula. Okay, now this morula, again it will undergo division and forms many number of cells. I am not showing all those stages because 2 cells, from 2 cells to 4, 4 to 8, 8 to 16, 16 to 32, 32 to many, and it goes on. Okay, I am not showing all those. Directly many celled embryo we have got. Now this many celled embryo, later it is getting converted to blastocyst. Okay, it will get converted to blastocyst. Now how does that blastocyst will appear? That blastocyst is something like this. Okay, it do have presence of, here I can show one layer of cells. Okay, with there is presence of inner cell mass. Okay, here there is a presence of cyst, that is cavity. The cavity which is present, for that we call it as cyst, and there is arrangement of cells. Cells have been rearranged themselves. The outer one we call it as, the outer ring cells we call it as trophoblast, and this one, these cells which are present here, for that we call it as inner cell mass. Right, so for this I shall show you in your diagram how it appears. See, this is the structure of ova, where many sperms have been reached. In that many sperms, only one sperm will be fused, so that zona pellucida will undergo certain changes. Then After this, right, you can find this diagram on page number 52 of your textbook. So this is the zygote, and this one is the ovum. Now that ovum, it is undergoing fertilization. One sperm has been entered inside and other sperms are not allowed to enter inside. In rare cases you can find that two sperms will enter inside, then you can get twins. That chances are also there, but in rare cases normally only one sperm will enter. Then after that, as soon as one sperm it has been entered, it leads for fertilization. After fertilization, what happens? It will start its mitotic division to form two cell stage. Each one we call it as blastomere. For blastomere stage, then comes morula. The embryo with 8 to 16 blastomeres, we called it as morula. And then you can find that cells, many number of cells, it has been divided and it has formed blastocyst. Now this is the blastocyst, which consists of one layer of trophoblast, and this one is inner cell mass, and this cavity is nothing but cyst. Because of presence of the cavity or a cyst, we called it as blastocyst. Okay, fine. Now this was the inner cell mass. Now the inner cell mass, what we have, that inner cell mass cells are also called as the above the inner cell mass, what cells are present of a trophoblast, for those cells we call it as cells of Rauber. I shall mention that as here, cells of Rauber. Okay, inner cell mass is present here. Above that, what we have find here, this is a trophoblast. Above the inner cell mass, the number of cells which are present in this region, for that cells, particular that region cells, we call it as cells of Rauber. And one more we are calling here is, as inner cell mass is present here, inner cell mass towards that side, what region is present? Okay, the side of the blastocyst to which the inner cell mass is attached is called as animal pole. This side we call it as animal pole, this side we call it as vegetal pole. Okay, we are using animal pole and vegetal pole. Animal pole, how we are saying animal pole? The side of the blastocyst where inner cell mass is attached, for that embryo, that part we call it as animal pole. The opposite of that we call it as vegetal pole. Hope you got this. This is an extra information which you don't have in your textbook, but you should know about this information. Right, we shall proceed further. Now when it has been reached to this blastocyst stage, where it will be, you can find in your diagram something like this. This is the female reproductive system. Ovary, it has been released ova. Fertilization took place in the ampla-isthmic region, and there is a mitotic division continues, and you can find it has been reached here. This is what it is, a blastocyst, which means this blastocyst is getting attached. It is getting attached where? That is to the uterine wall. Which uterine wall? We have given three uterine walls. One is the outermost one is perimetrium, inner one myometrium, and the innermost is endometrium. And for endometrium, this blastocyst will get attached. Okay, so how it will get attached, we shall see here. Now we have said that this is a trophoblast layer and another one is inner cell mass. We shall see what is the function of trophoblast layer. Trophoblast layer, it is useful for the getting attached to the endometrium. Okay, getting attached to the endometrium. Who is getting attached to the endometrium? It is this trophoblast layer. Okay, now what happens to the inner cell mass in future? Inner cell mass will develop into an embryo. Okay, inner cell mass will develop into an embryo. Now this one, when blastocyst it has been formed, now this blastocyst I shall show you here. This was the female reproductive system, right? I am showing only one side. Here we have shown it as blastocyst. Till here it was a zygote. It has undergone lot of division. As it has been continued its division, for a division, when zygote undergoes a division, we call that a division as cleavage. That simple mitotic division we call here it as cleavage. Commonly we call it as cell is dividing. Here when zygote divides, we call it as cleavage. Okay, so when zygote is undergoing cleavage, it is reaching to the blastocyst stage. Okay, now this layer which I have drawn, shall mention it as endometrium. Okay, so now blastocyst stage, when it has been reached here, then what happens? This trophoblast layer, it will start to form finger like projections, okay, so that it will get embedded in the endometrium. Endometrium also will undergo certain changes so that it will make a covering around it, and this process we call it as implantation. Okay, the process of embedment of blastocyst into the endometrium. So this endometrium, how it is getting embedded, that is because of the trophoblast. Trophoblast forms finger like projections. That finger like projections will appear like chorionic villi. Chorionic villi, which we have shown in an intestine, okay, in an intestine, or in yes, in an intestine we have shown that there are finger like projections. For that we call it as chorionic villi. Similar to that, this chorionic villi, they are formed, or finger like projections are formed, which are also called as chorionic villi. It will get embedded, it will get entered inside the endometrium and makes that blastocyst to get attached to the endometrium. That process is called as implantation. In a simple term, what is implantation? Okay, what is implantation? The process of attachment of blastocyst to the endometrium of the uterine wall is called as implantation, which means now this blastocyst is attached. Now it confirms that that individual is pregnant. Pregnancy is confirmed at this stage, which leads to pregnancy. Okay, so this is how it has been attached here. Now further we have pregnancy and embryonic development further, which means after getting attachment of this, further what happens to this blastocyst. About that I shall discuss in my next video. Okay, so hope you understood till here. If you have any doubts, any queries, please let me know. And I have to discuss about sex determination also, which I have said that sex will be determined at a fusion of male and a female gamete. So that also I shall discuss in my next video. I shall discuss about the sex determination, and then I shall discuss about the next one, that is pregnancy and embryonic development. Hope you understood this. If you have any doubts, any queries, please let me know and comment in the comment box. And stay tuned. Stay tuned. Stay healthy. Stay safe.